Okay, so we've learned about the three types of muscle, and we've learned about how muscles and joints connect and interact. Let's go into more depth with regards to the actual structures within muscle and muscle fibers themselves, and how these components of muscle, these molecules that make up muscle, interact when muscle contracts or relax. We'll come on to that bit. That bit is called sliding filament theory. Before we can understand sliding filament theory, we need to understand the actual structures within the muscle fiber itself. Now, the muscle fiber itself is made up of small repeating units, just builds on the previous explanation about muscles, okay? We finished that explanation with this small repeating unit within the muscle fiber itself. This diagram here represents one repeating unit. This is a sarcomere. A repeating unit from there to there is called a sarcomere. Okay? Now, in a sarcomere, we have some molecules. Two types of molecule make up a muscle fiber. We have actin, which is a long molecule, and we have myosin, which is also a long molecule. Now, the difference between these two molecules is the way we describe their appearance. The actin molecules are thin compared to the myosin, which are thick. Okay. Now, if we were to look at the structure in more detail within the sarcomere, we would see that the actin molecules form a structure whereby the myosin molecules overlap with them for some parts of the sarcomere. So, sarcomere is from here to here. We've got the actin molecules, which are these thin ones, and then we've got the wider myosin molecules here. We can see that in this region of the sarcomere, we only have actin molecules. These are thin. Under a microscope, this region of the sarcomere would look light under the microscope. We'd say this is a light band. It would appear quite light in comparison to the region next to it, where we've got actin and myosin in that same region. They overlap. Okay? And in fact, this region here, because of the presence of myosin, would appear darker if we were to view a thin section of muscle under the microscope. Okay? And then we've got this region to finish off the sarcomere, which is just actin. This would also appear as a light band. Okay. Now, these other parts of the sarcomere, they're not a physical part, but it's just a representation of identifying the middle of a region. If we look at this region in the middle, where there's only myosin, the middle of that region we call the M line. M for middle, M for the myosin only region. Okay. The beginning and the end of the sarcomere, we refer to that as a Z line. It's not a physical component, but if we were to look at many repeating units, we would see the end of one sarcomere and the beginning of another. Where one sarcomere ends and another one begins, that is called the Z line. And during a muscle contraction, the Z lines can become closer together and further apart, which we'll learn a little bit more about later. Okay, today is just about the structure of that repeating unit, which is the sarcomere. So we have our Z line, which is the beginning and the end. We have our M line, which is the middle of the myosin only region. M for M line, M for myosin, M for middle. All the M's go together. We've got the I band, which appears quite light. And if we look at the letter L and the letter I, they're very similar. So remember, the I band is the light region. Just by linking the appearance of the I and the L, they go together. Okay. If we were to take this sarcomere and look at it as a cross section, and what a cross section means is we have the sarcomere like this, we cut it and then we look down it. That's a cross section. So if we were to take the sarcomere from the page, turn it around, slice it, and have a look at what we can see, we would see, if we cut it here, we would only see the ends of our myosin molecules because those are the only ones that are there. If we were to do a cross section here, we would see the ends of our myosin molecules as well as our actin molecules. Okay. So that's the structure of a sarcomere. We need to be very good at understanding the different bands in the different regions and what their names are. Okay. Quickly going to go on to, no, that's it. I just want to concentrate on the structure of the sarcomere.